This is a short video abstract about a scoping review published in Brain Communications. The focus of this review was the arrangement of white matter fibers within the cerebral gyri, which are key structures in the brain. A cerebral gyrus comprises an external layer of cortex and an inner white matter core. The architecture of this core has specific features that make it distinct from deeper brain regions. Limited externally by the gray matter that covers the top of the gyrus and the neighboring sulci, the gyral white matter comprises a mix of fiber populations with multiple directions and destinations. This fiber colocalization is key for the functional role of each cerebral region and is critical to clinical manifestations in case of parenchymal damage.
Our scoping review presented key concepts and primary source of evidence. They shed light on the complex organization of fiber pathways within the gyral white matter. The investigation involved a comprehensive search across various databases using keywords related to the architecture, anatomy and morphology of the white matter fibers within the gyra. After extensive analysis, we incorporated findings from the 73 relevant publications. The presence of densely packed fibers with multiple crossings, the proximity to the cortex, and the existence of interregional and inter-individual variations make the task of depicting this microanatomy extremely challenging. This is the general pattern in which these pathways are distributed based mostly on the seminal works on non-human primates. Various types of fibers connect adjacent and the distant gyri, including associations, striatal and cord fibers. Fiber dissection and radiological methods like magnetic resonance-based diffusion imaging have also been used to study these connections and their three-dimensional arrangement. The literature review highlights that the white matter organization in the gyri is not uniform and varies across different regions. An important source of variation is that the proportions of different white matter subcomponents vary in the brain gyri. The main patterns as a function of brain topography were explained in the publication and illustrated with typical examples. We propose classifying human gyral white matter structure based on these patterns to synthesize this information. Other situations are the convergence of short association bundles, often in confluent gyri or intragyral short association bundles. While progress has been made, comprehensively studying gyral white matter remains challenging due to its complex nature. The topic holds, however, paramount relevance for both fundamental and the applied neuroscience.
Clinical applications of gyral white matter understanding range from neurodegenerative disorders to surgical planning and potential insights into psychiatric conditions and cognitive processes. In conclusion, the gyri of the human brain are key topographical arrangements of the convergence of white matter tracts with an overall organization that appears similar to that of non-human primates. However, current data highlight many regional adaptations bearing enlightening morphofunctional correlations. The most drastic changes in the general pattern are fundamentally due to differences in the proportion of fiber populations in the gyral white matter. Variants are numerous, but most often fall into one of these categories: prominent association fibers, prominent projection fibers, prominent projection and commissural fibers, and prominent short association fibers. In order to better understand this complex hodotopical organization, a deep understanding of the three-dimensional colocalization of white matter fibers in the gyrus and their origin in the brain is paramount. We underscore the clinical importance and the potential of gyral white matter studies in advancing our understanding of brain connectomics. For further details on this research, please refer to the full article in Brain Communications.